Hi, my name is Scott Rock. Today I'm going to be presenting on a protein called MMP-9 and how it relates to hearing loss when studied in the mouse model. And so in order to study hearing loss, we really do have to spend some time and talk about how hearing functions in a normal, healthy system, right? And so I know I just said we'd be talking about the mouse model and I put a picture of the human ear up on the slides, but there's really not a whole lot of difference. And the reason for that is that these are evolutionarily inherited traits. They're preserved. And so whenever we find clinically relevant material in a mouse ear when it comes to audiology, it can also be applied generally to the human structure as well. And so that's very important for these studies in particular, because many of them were studying mouse models with the goal of understanding something more about human hearing loss. And so let's just orient ourselves to how this diagram is set up and what regions of the ear we're talking about. And so we've got the outer ear that ends at this eardrum here and includes this whole outer structure, everything you can see in the ear, right? And this is going to be exposed to the elements. It is not sterile at all. And then we've got the inner portion. So we've got the middle ear right here, which is going to include the eardrum and these three bones called ossicles. And then we've got the inner ear, which is going to include this big purple portion called the labyrinth. And the most important part of the labyrinth today is this curly snail region called the cochlea, right? And so let's start to get into how hearing works now that we know what this diagram is really showing. And so sound is going to enter the ear from the environment, right? And it is going to be funneled into the ear canal by this funnel shape on the outside that we've all got called pinna. And so it is going to hit the eardrum, right? And so this eardrum is going to vibrate, also called the tympanum. Its only job is to really vibrate in response to noise hitting it. And what that's going to do is it's going to vibrate these ossicles. These bones are kind of touching this eardrum, and they're going to amplify this noise. They're going to vibrate even harder in response to this noise. They're all going to shake together, right? And so what's going to happen next is something really, really interesting having to do with this purple structure. Particularly the cochlea down here. And so within the cochlea has its own fluid in it called endolymph. This whole thing is kind of like a water balloon, right? And so think of a glass of water placed on a counter during an earthquake. It's going to shake back and forth, right? And so this, you can kind of think of it as a glass of water during an earthquake. And so you've got these ossicles and they're vibrating very hard, very nearby. And so that liquid inside the cochlea is going to shake and turn. And the cochlea has some very interesting, very intriguing cells in it called hair cells. So these hair cells are sticking up off the membrane. And whenever that fluid shakes, they're going to be disturbed too, right? And so they're going to lean with the fluid, right? And they're going to register those changes in orientation. And whenever that happens, a nearby neurotransmitter is going to get the signal and it is going to interpret it as electricity. And the auditory nerve is going to collect these signals and send them off to the auditory cortex where you will hear them as sound. And all of this happens in a fraction of a second, right? So this is obviously a very complicated system to study, right? And it's clear to most that this is just an oversimplification of what hearing is. This is just an overview. But we're going to be focusing on a very particular structure today called the cochlea. And the cochlea is this portion down here of the labyrinth it is heavily focused on hearing, right? The upper portion of the labyrinth is going to be involved mostly in balance, and so it's irrelevant for this talk today. But down here, this is really where the magic happens when it comes to hearing. So we're going to be zooming in even further to some of the microstructures of the cochlea, of how the cochlea kind of maintains itself. And so this is kind of a complicated diagram, and we don't really need to understand all of it. We just need to understand that there's kind of a line right here dividing the inner cochlea from its surrounding environment, right? And so, but right in here, we've got this strange region called the stria vascularis. And the stria vascularis is going to be responsible for delivering some of the nutrients that the inner cochlea needs to maintain itself, right? And so this is a sterile region. And it is not able it has quite a few cells that need products for their metabolism, but they don't have any way of getting it themselves because they don't have blood access going straight into this region. So instead, the best they can do is they deliver capillary loops as close to the sterile structure as possible, right? So the stria vascularis is really just this structure of capillary loops getting as close as possible to the cell membrane of the inner cochlea. And so once it gets close, what's going to happen is that some nutrients are going to diffuse across those membranes, and these cells are going to take it up, right? And so we're going to zoom in a little further into the capillary structure now and talk about some of the anatomy of those capillaries. And so here we can see this is a blood vessel, but it applies for our purposes that these capillaries are going to be pretty tightly bound to each other, right? And so we've got this lumen in here where things are going to be flowing downstream. And here on the edges, we can see that there's kind of this cellular structure where these are bound together. These big flat cells are going to be bound together to each other with something called a tight junction. And so these tight junctions, they're going to keep these cells as bound together as possible, right? If you think of it as they're kind of linking elbows, linking arms, that's a good way to think about it, because their goal here is kind of to keep everything in the capillary that has to stay in the capillary. And so maybe there's some pores here that are letting some very small nutrients get out. But by and large, larger molecules should not be able to permeate this barrier. And these cells are also going to be attached to this basement membrane, shown here, that's the semi-clear bit, where these endothelial cells, they're also going to be adhered to each other and adhered to this basement membrane. And so it's clear to us that this whole system of kind of a border control, of keeping everything inside this lumen that needs to be in there, it has quite a lot to do with cell adhesion. And so let's just take a closer look really quick at cell adhesion, and specifically what kinds of cell adhesion are going to be relevant for us today. And so we have five different types of cell adhesion on this diagram here. And so we're going to focus today on this one up here called a tight junction, and on these two down here called focal adhesions, right? And so imagine that these are cells in a capillary wall. We've got some cells back here, and maybe this is a lumen, and there's material flowing downstream this way, right? Let's say we have some molecules floating around. And because they're flowing at a pretty high speed, maybe they get caught right between these two cells, right? Do you really want them getting all the way down here and, maybe permeating the basement membrane and getting into whatever is down here, right? I wouldn't, right? And our cells don't either. And so that's why they have tight junctions. So these are very strict fences, kind of, right? And so what they're going to do is they're going to stop any small lost molecules from getting too far into this cleft. They're going to just kind of redirect them back to the lumen, back to where they need to go. And so this is the system that we're looking at. And so this is a much more simple view of that system, right? It only has what we really need to know for this model. And it is a very unspecific, right? You get nothing in this system about where we are in the body, what kind of applications this has yet. All we have is just some cells that are attached to a basement membrane and are attached to each other. And so you can see these focal adhesions that I'm highlighting here in red down at the bottom. So they're attached pretty strongly to this basement membrane here, right? So imagine this is a cell, this orange thing, and it is linked together with this cell, with its buddy, with its neighbor, by ZO-1, a protein called ZO-1. And that is one of several proteins involved in a tight junction, but it's the one we're going to be studying in our main paper. And so imagine for a second that these blue bars that I'm highlighting right here, that these are kind of like string linking these together. They're kind of tying them together. Well, if that's string, then we've got to talk a little bit about what MMP-9 is, which is the best metaphor we can use is a pair of scissors, right? And so MMP-9 is going to come in between these cells, and it is going to kind of cut ZO-1 off. It is going to sever this thing. It's going to snip it, right? And it might do the same down here too, right? And so imagine for a second that we've got a cell right here, right? And that it is attached to a basement membrane, and that it's attached to its neighbor. And then MMP-9 comes in and it snips the string. Well, now it's not attached to anything. And imagine that this is a lumen, and that there's liquid flowing downstream, this is going to be picked up and carried away. And now it has lost some of its differentiation, and it's going to live a nice long life somewhere far, far away. But in the meantime, you've got this gaping hole right next to your basement membrane, which is pretty flimsy on its own. And the purpose of that, this is actually an evolutionary structure. I know I've been talking about this as if it's a pathology, but this is definitely something that your body does and does often, is that you have opened kind of a door for any immune molecules that need a really quick way through this basement membrane to kind of get to the other side and fight off an infection maybe, right? And so MMP-9 is really, it's kind of a bulldozer to kind of break through this membrane and allow some travel between a strict membrane like that, right? And so we can see from this diagram that this is a very unspecific kind of program, that you could apply this in a lot of different body systems, a lot of different tissue types. And because it has such a wide variety of uses, it also has quite a few implications in pathology and in disease. The most well known is going to be cancer metastasis. And so when cells break off like this and travel downstream, it's not difficult to imagine how that might be implicated in cancer spreading, right? If failure in cell adhesion implicated by MMP-9 is induced, then maybe a cancerous cell makes its way to somewhere else and takes root. But it also has quite a few of other conditions. Heart failure, stroke, ocular hypertension, and inflammatory conditions such as arthritis are all heavily linked to MMP-9. And some of these you can kind of see how the system might play in, but some of them are a little less obvious and take a little more thinking to understand how this basement membrane string and scissors approach might play in, right? And hearing loss is one of those. It's not immediately obvious. And so we're going to spend quite a bit of this talk discussing how MMP-9 and Z0-1 play together to cause hearing loss. And so we're going to be spending a lot of time on this paper called "The Lipopolysaccharide Disrupt the Cochlear Blood Labyrinth Barrier by Activating Perivascular Resident Macrophages and Upregulating MMP-9." And so that's quite a long title, and we're going to refer to it as Zhang et al. 2019. But what it's studying essentially is this relationship between MMP-9 and a disease called labyrinthitis. And so labyrinthitis is going to be a condition that is inflammation of the inner ear. So we talked quite a bit about that cochlea and that microstructure, the stria vascularis. And so that's where this is going to come in, is in the inflammation of these small microtissues in an area that's not really supposed to have access to a lot of the general circulatory system. And so we're really looking at this model of MMP-9 breaking down basement proteins, basement membranes, in this way that we've represented here, but in areas where it's not necessarily supposed to do that quite a lot. And it's going to kind of bust down doors into regions that are very sterile and maybe even introduce some agents that could cause inflammation in places where there's not really supposed to be inflammation. And so it is going to focus on the interplay between these two proteins, MMP-9 and Z0-1, because they're often linked together when we're talking about cell adhesion. All right, and so let's take a little bit at and get some of the terminology out of the way that we're going to need for this talk. One of the most important terms to understand for discussing the condition labyrinthitis is the blood labyrinth barrier. And so you might have heard of the blood-brain barrier before, and the blood labyrinth barrier is about the same idea. The labyrinth, which is this purple region in the ear here, it is not supposed to really have access to much of the outside circulatory system, right? And so it has a very strict kind of clearance process in order to get in this area, which is kind of what you think about with the blood-brain barrier, right? It's kind of like a border control, keeping things in and out. And so that is definitely what we see here. And what we see here is a diagram showing the blood labyrinth barrier. And so you have some endothelial cells right here, and we've seen this already in terms of capillaries. So this is nothing new to us. We've got tight junctions and they're stuck to a basement membrane, right? And so these cells they're being supported by a cell called a pericyte, right? And that's going to be up around the side. And then you've also got these cells called perivascular resident macrophages. And these are what's actually going to excrete the MMP-9. So we've been talking about the MMP-9 as if it just exists in this system, but it's actually excreted mostly by these macrophages. And so these macrophages are kind of your border control agents. And so they're going to police what gets in and out. They're immune cells primarily, right? And so if you think of this as one side of a border and this is the other side of the border, PVMs are going to really be responsible for determining what comes in and out. But equally important is going to be how accurate, how tightly bound these cells are. Because if they're letting things out, then the PVMs themselves are not going to be particularly helpful. All right. And so let's talk a little bit about what they were expecting to see in this experiment. And so they're introducing something called LPS, right? This is in the title, lipopolysaccharide. And so LPS is going to be an inflammatory agent because it exists in the outer wall of bacterial cells. And so anything that sees it is kind of going to set up alarm bells as that it is a sign of an infection, right? And so if LPS is going to trigger disruption of the blood-labyrinth barrier, if that's our hypothesis, that LPS is going to cause some hearing loss through breaking down this blood-labyrinth barrier, the first thing is you have to show that there's some hearing loss, right? Because that's going to be the linchpin of the study. If that doesn't happen, then you can't really prove that anything else had any relevance, right? But at the same time, once you have proved that there's significant hearing loss caused by the lipopolysaccharide, the inflammatory agent, you then have to show that it has to do with regulation changes in MMP-9 and Z0-1. Because there's a lot of different things there's a lot of different things implicated in inflammation. You have to point out specifically that it has to do with MMP-9 and Z0-1 regulating these borders, right? And then on top of that, you will have like a vague correlation there. But to really hammer it in, you do need to demonstrate that the regulation between MMP-9 and Z0-1 has to do with the blood labyrinth barrier and with those tight junctions falling apart and really letting things in, being defective in blocking some of these agents from passing this barrier. And so those are going to be the three kind of linchpins to this study. And if we fulfill all of those, we can successfully say that we proved that MMP-9 can cause hearing loss. And so let's go a little bit into their methods and talk about how they group this up. And so we broke these mice up into two groups. One of them received lipopolysaccharide, our inflammatory agent, and one of them received saline, which is not going to inherently cause any kind of inflammatory response. And what we did is we injected them transtympanically. That means across this eardrum here into a region that is not really supposed to have access to the outside world, right? This is the sterile region of the ear. And we let that response proceed for 48 hours. And the reason we had to inject both of them with it is because injecting across the tympanum is going to be an inherently inflammatory process in both of these mice, the one that receives the saline and the one that receives the lipopolysaccharide. And we don't want to compare the one that receives the lipopolysaccharide to something that didn't actually have any inflammatory response at all, because the way we're introducing it is inherently a bit invasive, right? And so we let this response proceed for 48 hours, 2 days, just to let all of the inflammatory processes proceed and let its infection response happen. Once that has proceeded, we're going to test whether or not these mice have any hearing loss, because that's our first big step. That's our first big linchpin. We have to show that they have some hearing loss. And so the way we're going to do that is with auditory brainstem response testing. And so that's going to be, mice are going to be under anesthesia. They're going to put electrodes on their brain, and they're going to play different sounds and see if their brain responds to it with an EEG, right? And so this is a very objective way of measuring hearing loss. If we were doing it in a human model, we might do a more standard audiological test. But because mice can't really tell you if they hear something, this is the best way of getting good data on hearing loss in an animal. And so then we're going to sacrifice these mice and harvest them. And we're going to do 3 things with that material. We're going to test the QRT-PCR, the levels of mRNA in these cells in the cochlea. We're going to do immunohistochemistry on the cells of the cochlea, looking at different proteins and where they might be, where they're found, and in what amounts. And then we're also going to look, just to kind of really hammer it in, we're going to take a very close look at this inner ear microstructure with a transmission electron microscopy, right? With a TEM structure, right? And so these are the 3 things we're going to do. And so our first big thing is we have to show that there was hearing loss. And so this is what this graph is. And so we can see here that this is titled threshold shift. And so what does that mean? Threshold shift is kind of like the absolute value of hearing. So the higher the number here on the y-axis, the more hearing loss you've experienced. And so what you see here is that these squares are the treatment, this higher bar is the treatment. And so they are ranking anywhere from 20 to 50. And that is a really wide range, but it is significantly more than this control here. That is quite a lot of decibels to lose. And so you can see here that they have lost 20 to 50 decibels of range depending on the frequency. That's a pretty significant amount of hearing loss. And then the control group had 10 decibels or less of hearing loss. And that is mostly due to just the fact that they were the introduction of the saline itself was a bit inflammatory, right? And so once we've got that established, that the mice did have an actual immune response that caused hearing loss to the LPS. Now we have to talk a little bit about what that immune response entailed. And we have to implicate these two proteins we've been focusing on, MMP-9 and Z0-1. And so let's just take a look. Let's just remind ourselves real quick of where we are. And so this is the inner region of the cochlea right here. And so we are still in that small structure called the stria vascularis, right? And so this is a close-up of some of these immunohistochemistry tests. On the top row, we're testing for MMP-9. On the bottom row, we're testing for Z0-1. On the top column, we're looking at the control. And on the top right, we're looking at the ones that are responding to the inflammatory agent. And so let's look first at figure A here, the control of the MMP-9. So the blue and dappy, that's all of your nuclei. And so I can see that there's a lumen right here, right? And so there's maybe some endothelium up here, and maybe a basement membrane right here in red. I don't really see a whole lot of MMP-9 though. If MMP-9 is going to be illuminated in red, I'm not seeing a whole lot of it in this control. But then we go over here to the one that was treated with the inflammatory agent, and it is really lit up, right? And so I can see there's definitely MMP-9 right here along this lumen. And then as well along this basement membrane, there is quite a buildup of it as well. And even in the middle, in these cells in the middle, you can really see quite a lot of red. And quantitatively, you can see to the right in figure C, that really significant increase, basically doubling the amount of MMP-9 we're producing. And so now let's go down and look at the Z0-1. Now that we've established that MMP-9 is going up in response to this inflammation, Z0-1, what is Z0-1 doing? So we've got the same system. Here's the lumen, right? And here is kind of that lining around the lumen, and you've got this endothelium tissue right around here, maybe basement membrane right here. And there is quite a lot of Z0-1. This is lit up in red, and so there is quite a lot. And if you think of Z0-1 as the thing that's holding those tight junctions together, that kind of makes sense, right? And so you'd want a lot of string kind of holding these cells together and in a typical healthy system. But then you induce this inflammatory response with the LPS, and it looks quite a bit dimmer in figure E. I can't see quite as much Z0-1. I can see still quite a significant amount here in the back, but towards the front, especially along this basement membrane, this first basement membrane, there is not a whole lot. And that can be shown quantitatively in figure F, where you see that it is not as dramatic as the MMP-9 increase, but there is still a significant decrease in the amount of Z0-1 that's holding these proteins together. And so what we have to ask at this point is do you think that this is enough to show that MMP-9 is causing hearing loss and labyrinthitis? Because I don't think it's enough. So at this point, we've got that we've caused hearing loss. We have caused hearing loss with the introduction of an inflammatory agent. But we haven't really proved that MMP-9 is directly involved. We haven't tested any other proteins. We don't know. This could just be an artifact, right? And so the authors knew that, and that's why they did some further testing, right? And so what we see here, and this is the mRNA levels. This is our QRT-PCR. And so we're looking at whether or not these cells have changed how much MMP-9 they're producing at that level, right? And so this is a bit more of a significant level because one mRNA can produce very many proteins, right? And so we can look here. This is obviously the linchpin of this whole graph right here. We're going to ignore these two for brevity's sake. They're not quite relevant to this talk. But MMP-9 here, this is the normal level. And here at the at the highest it gets in response to inflammation, it is almost 6-fold increase. That is an incredible amount of mRNA to produce in response to an infection. And Z0-1 has the opposite effect. It is going down in response. And so this is the same thing we've seen here. And so this is absolutely supporting that theory we have that MMP-9 and Z0-1 are working together, that they're behaving together, and that this is not really an artifact, that this is a function of evolution. And so what would you want to know at this point is, how is this related to the capillary leakage, right? And so at this point, we have shown that there is some, statistically relevant increase of MMP-9 and statistically relevant decrease of Z0-1. But how does that relate to this idea of the capillaries leaking and maybe causing some inflammation? Well, the researchers were very obviously attuned to this, and they wanted to show clearly that MMP-9 and Z0-1 were related to capillary leakage, because at this point they hadn't shown that capillary leakage was even occurring. So what they did in this study, right, and these 4 panels here, is they introduced something called FITC dextran into these capillary structures. Now, FITC dextran, that's a sugar, right? And so that sugar is not going to be able to permeate these barriers, these capillaries naturally. And so by injecting it, you're really just kind of getting an idea of where the borders of the capillaries are. And you can see very clearly that this is kind of just like, it looks almost like an X-ray of these capillaries, right? You don't really see any of this this lit up fluorescent FITC dextran anywhere other than in these capillaries. And this is what you'd expect to see in a typical model. But then you look down here at the one that's been treated with the LPS that has had the response to inflammation, and you see something very, very different. And so we've still used FITC dextran to light this up, and it should not be able to pass an intact capillary barrier on its own. But these capillaries are kind of nowhere to be found. So I'm drawing some of these barriers that I can see with my eyes, and they don't look anything like capillaries. And so it is pretty clear looking at this that there has been some leakage. This looks like a popped water balloon right here. But this whole structure right here, it is pretty clear that these capillaries have lost some structural integrity, and they are letting sugar through these capillary barriers. And so if we just orient ourselves really quick to this diagram on the right that we saw before, that this is a function of those tight junctions breaking apart. So we can think on this side, if these tight junctions were intact, that's where the FITC dextran is just going to float around here. But on this side, if we have big gaps in everything, it's just going to flow right out. And so now that we've established that there is some capillary leakage, we've got a pretty compelling model already. We've established that there is hearing loss in response to an inflammatory agent in a place where really the inflammatory agent shouldn't be able to get on its own. And then we've established that a protein that is known to dissolve basement membranes was very active in that region. We've established that capillaries leak when exposed to LPS. And so this is already a very compelling model, but the researchers really wanted to hammer it in and show very clearly what is going on. And so they did transmission electron microscopy, where they took a closer look, as close a look as they could, at some of these tight junctions in the middle ear, in the inner ear, sorry. And so you can see right here, and so here's our control at the top. Here's the lumen of the capillary, and it can be a little bit difficult to interpret these diagrams. So let's just take a minute to orient ourselves to this being the lumen, this light area being the lumen, and then zoomed in up here to the right, we have this close structure all around. We're looking at the endothelial tissue that is separating this capillary from the stria vascularis around it, right? And so we've got this tight junction, we've got this endothelial cell, and you can kind of see the borders of the cells, but it's mostly just based on the topography. And so you can imagine that these are kind of held together with these tight junctions. And you can very clearly see that there's a basement membrane between them, that this is where they're being anchored to, right? And so this is what you would expect to see in a healthy, functioning stria vascularis system. But then you look down here, and I know this has been the model for the entire way, is that we're kind of throwing a wrench in the gears of some very intricate, beautiful systems, and we're not stopping now. Is we've got this capillary here, and I've kind of traced the edge, and it is pretty much collapsed, right? This is not exactly the structural integrity you would expect from a capillary, and this is definitely not the structure you would expect from an endothelial tissue surrounding it. But then we're going to zoom in a little further on just one region to illustrate how this has fallen apart. And so here's the lumen right down here, this portion right here. And then it's very difficult to see the delineation of the cells anymore, but let's say for the purposes of illustration that these are 3 cells right here. Here is the basement membrane. So these cells may or may not still be anchored to each other. There is definitely some empty space elsewhere, but this basement membrane is definitely not anchored to them. There is empty space here. This is the same color as the lumen. And so there has been a collapse of this structure. And so anything can go anywhere at once now, really. And so that is just a quick overview of how that system works. And so let's just quickly summarize and put this all together and talk about what the study means in terms of the wider world, right? And so we introduced this inflammatory agent called LPS, and that upregulated MMP-9 and downregulated Z0-1, meaning that we got more scissors and less string, meaning that more cells are going to break off of their basement membranes. These tight junctions that keep this border control system is going to collapse, and those capillaries are going to leak into places that maybe they shouldn't leak, right? And so that is going to be a break in sterility, specifically in this cochlear structure, the stria vascularis. And what that's going to cause is inflammation in the cochlea. Those molecules are going to be introduced to the cochlea, and that is going to cause a widespread inflammatory reaction called labyrinthitis. And labyrinthitis very often leads to hearing loss because of the fine structures in this system that need to respond very carefully to their surroundings, right? And so at this point, it is important to discuss whether or not this system is generalizable. Because there is a bit of an experimental concern here. MMP-9 in a typical living system is not really going to be upregulated for that long. It's a very transient molecule. You know, if you've got this wall here, it's going to break a hole in it, and then something else is going to come in and repair it. But because that is generally in response to a whole kind of viral or bacterial molecule, right, being introduced, that is the purpose of that is to introduce a macrophage that can go and eat it. But in the case of LPS, they're very small molecules. They can kind of get stuck in the nooks and crannies. And so what you might have is some of this like chronic inflammatory response that may or may not really be applicable to a human system. And so it's really just something to keep in mind. But at this point, just to wrap up the talk, I think it's important to kind of zoom out and talk about how MMP-9 can cause hearing loss in other places, because we've been talking about labyrinthitis, which is a major cause of hearing loss, but it is not the only major cause of hearing loss. And so we remember that system of the hearing, how it's very complicated, how we oversimplified it to like a 2-minute segment in this talk. But there is really quite a lot of steps involved. In MMP-9, we saw how generalizable that system is. It can go anywhere where there's a basement membrane. And so it is really plausible that MMP-9 could cause hearing loss in other places as well. And so we're going to really quickly talk about a couple of those places because it does indeed cause hearing loss in other places. And so we kind of stopped at this model where the auditory signals are sent off as electricity into the brain, but where they go is the auditory cortex around here in the brain. And this is where MMP-9 has quite a lot of implications, right? And so we're just going to really quickly go through some of them with the idea that extracellular matrix is kind of everywhere, and MMP-9 is pretty much involved anywhere there's extracellular matrix, especially when it comes in terms of basement membrane. And so the brain has quite a few different ways to account for hearing. The auditory signal travels to a few different places, right? And so your first big implication for MMP-9 is that it is involved in age-related hearing loss. And so that's just one of the ways that it can cause hearing loss, is that it breaks down extracellular matrix in the way that it is really expected to in a kind of normal environment. But then because the brain is aging and it doesn't really have as robust a response anymore, it doesn't go back in and repair that damage. And over time that can build up and that can introduce things and cause some neuroinflammation. And so that is one way that MMP-9 can cause hearing loss, but specifically age-related hearing loss. And then another way that I think is particularly relevant to this talk is it can cause meningitis, of which a really common side effect is permanent hearing loss. And so you think about the blood-brain barrier, right, as being kind of this border control that we just talked about for a long time in terms of the stria vascularis. And so what you have sometimes in the blood-brain barrier is of course, that's a more complicated system, but the same application of MMP-9 going in and, the scissors and string, cutting off some cells, breaking open a spot for an immune cell to come through, but also maybe making that more permeable for other inflammatory molecules that can pass through that. This is also quite relevant. So if you have an infection in your blood and maybe MMP-9 malfunctions in the blood-brain barrier, you could get meningitis from that because it'll be introduced to your brain. So that's exactly the same system we've been talking about this whole time, right? And so that is another way that MMP-9 can cause hearing loss related to the neuronal structure specifically. And so the last way we're going to discuss is this one I have on the screen here, which is audio cortex neuroinflammation, which is a very specific condition that has to do with the dissolving of perineuronal nets, which are just a type of extracellular matrix in the brain. And so they're also stabilized by basement membranes. And so MMP-9 can go into these places and they can cut them up just the same way we've been seeing the whole time. But because those extracellular matrix cells provide some very essential signals for your neurons to fire properly, it can hurt the way you hear. And so your signal, that auditory signal, can go through all those steps to get to the brain. But if the perineuronal nets aren't there to support the neurons interpreting those signals, you're also going to have hearing loss from that as well. So what really is the take-home message from this? And so when it's functioning correctly, MMP-9 is a really interesting, charismatic protein that has such a simple function that can be applied to such a wide range of systems, right? But because it's applied to such a wide range of systems in helping fight infection and restructure some injured tissues and trigger epithelial to mesenchymal transitions, it is also implicated in quite a few pathological conditions as well as including hearing loss. And so when it functions incorrectly, it destroys extracellular matrix and allows intruders into areas that they really shouldn't be, like the labyrinth. And so this is a great example of a well-adapted system being applied to areas in the body where it's not really meant to be placed, right? And so it's behaving inappropriately. And so that is really an example of just a very simple function that is just applied in like 100 different ways across the body, right? And I think that is a really important thing to understand as biologists, is that there are very simple systems that can have really wide-reaching consequences that the more you study, the more they just get more complicated, right? And so thank you for coming and thank you for watching. And this has been great. Thank you very much.